Well, let's move to the next lecture. We will talk about staphylococci. Let I show you this flow again. So the last three lecture 9, lecture 10, 11, we done on general characteristic of bacterial pathogen. You learn cell structure, cell function, pathogenesis, metabolism, antibiotic, and how bacteria resistant to antibiotic. Okay, now we learn some usual bacteria, general bacteria. Okay, we classify bacteria into two main groups, gram-positive and gram-negative. Gram-positive, if, well, whatever, in the lab, doctor sends specimen, you need to culture. Once you culture, that's spent overnight. Day after that, scientists supposed to smear the colony on the culture to observe what gram. And if you found this gram-positive cocci, arrangement of cells is very helpful. You probably see arrangement of cell as clustered like this. Then we are talking about staphylococci, okay? So staphylococci, by the name, staphyli mean grape, like bunch of grape. This is fake color, okay? But coccus mean berry. Staphylococci mean like it is cocci arranged like a grape shape. Classification of bacteria based on, we have a society of microbiology that make agreement of the classification of bacteria and make a book, a manual book called Berge Manual of Systematic Bacteriology. Now we have second version. The first version said Staphylococcus grouped together with the other genus in the family Micrococcaceae. Micrococcaceae is arrangement in 4 cells, not a bunch of cells like Staphylococcus, okay? But the new classification, Staphylococcus moved out from this family, form their own family named Staphylococcaceae in the phylum Firmicute, okay? Firm is means strong, cute is means skin. They have strong skin because they are gram-positive, okay? Major classification of Staphylococcus, when I say Staphylococcus or Staphylococci, I mean in general, not point to any species, okay? They are gram-positive cocci, okay? Like the gram stain like this, the grape-like shape, you can see grape, okay? They comprise so many species, but most thing that it probably help you to remember almost all Staphylococcus, I mean 95% up. Let's say this is the characteristic. They show up with catalase positive. So they have catalase enzyme. Still remember, catalase enzyme is the one that convert hydrogen peroxide to water. Okay, that make Staphylococcus mostly aerobic or facultative anaerobe for some species. They can change the pathway inside from the aerobic respiration to use fermentation. Okay, they cannot move. They cannot process flagella. They cannot produce spore. That's characteristic. Okay. But you need to remember catalase positive. Okay. The colony on TSA, tryptic soy agar, they show up with the, well, textbooks say deep yellow. Some textbooks say the butter color, but the yellow butter, not white butter. Okay. It is normal flora on human skin and mucous membrane in your nose, in your throat, in vagina also. That's, we can find staphylococcus. Okay. One important ability of staph is ability to hemolyze blood. They destroy red blood cell. Okay. Once you grow the Staph aureus, sorry, not Staph aureus, any staph, particularly aureus on blood agar, you can see there are clear zones around the colony on blood agar because they release some enzyme to digest the red blood cell. Okay, that is the staphylococcus, and also they have ability to clumping the plasma. Do you know what is the plasma mean? You learn immunology, still remember? Plasma and serum.
Blood serum.
How different?
Blood serum has soluble proteins in it.
No.
Or is that the other way around?
Serum, when you draw blood from human, okay, the blood start clotting if you do nothing, okay? Once they clot, the solution, leftover solution, is turned from red yellow. You might see the clumpy of clot is red and the solution is yellow. That one is everything inside the solution of the blood except blood clotting factor. Okay, so plasma is serum plus blood clotting factor. So this blood never clot it. So all component of the blood still remain in the plasma. Okay, some species of Staphylococcus can make the plasma clot, but some are not. Okay, this is the key thing that you learn later. They produce so many extracellular enzymes and also so many type of toxin. Okay, there are, like I said, so many species, but in this lecture you need to learn 3 species: Staph aureus, Staph epidermidis, and Staphylococcus saprophyticus. 3 of these. The biochem test for Staphylococcus, one is detecting catalase enzyme. The catalase enzyme convert hydrogen peroxide to water. Okay. So you just drop hydrogen peroxide, 1 drop, 2 drop, and pick up the colony of Staphylococcus and smear on the hydrogen peroxide drop. If you observe a lot of bubble within 2 or 3 seconds, this is positive because they generate water and oxygen. If you see nothing change, that means negative. So Staphylococcus are catalase positive, need to remember. So coagulase enzyme is another type. Coagulase enzyme from Staphylococcus aureus, this is mean aureus, can react with prothrombin. Prothrombin is blood clotting factor. And then they change prothrombin to staphylothrombin. This is protease enzyme. This protease enzyme will digest fibrinogen to fibrin. And fibrin, a lot of fibrin make plasma clot. If you put plasma inside the tube, and inoculate Staphylococcus with this tube is Staph aureus. This tube with the other staph, you probably see it is clot, like make the protein inside the plasma clumpy to like a ball. That's the key laboratory test. So let you remember easier. When you get specimen culture on blood agar, you observe yellowish colony or white colony, that's okay. Perform gram staining. You might get you need to get gram-positive cocci arranged in cluster. Perform catalase test positive, it is Staphylococcus. Okay? If negative, you learn next week. So this week, when you say catalase positive, you can say it is potentially Staphylococcus, but you don't know which Staphylococcus until you perform coagulase test. Positive coagulase show up Staph aureus. Negative show up the other staph, okay? Probably Staphylococcus epidermidis or something else. Only aureus are positive, okay? Right. So let's start from Staph aureus. The characteristic of aureus look like general staph. Normal flora of skin in the nasal tract. Arranged in clusters. Cell size is about 1 micrometer. Non-motile, non-spore forming. Okay. Just few strain can produce capsule, but mostly not. In TSA, tryptic soy agar, you probably need to culture on 37 degrees Celsius at 7.4, 7.5 pH. They are arrow on facultative an arrow. You see the yellowish colony. The colony is quite small colony on TSA. That is Staph aureus. But if you culture on blood agar, you see hemolysis. Hemolysis by clear zone around bacteria. The clear zone like this, we call beta hemolysis. Okay? But no hemolysis is possible. Mostly are beta hemolysis. I would say hemolysis or not hemolysis is the main, it's not important to classify, but just let you know, mostly beta hemolysis. Sometimes we culture Staph aureus on McConkey agar. So this is the thing that you need to remember first. You learn a lot in E. coli. McConkey agar is a key medium in E. coli. McConkey agar contains some substance that inhibit gram-positive bacteria. So most of gram-positive bacteria cannot grow, okay? Because they have crystal violet inside McConkey agar. The crystal violet inhibits cell wall synthesis. That's make gram-positive bacteria cannot survive, okay? But Staphylococcus aureus tolerate those things and show up the yellow, not yellow, sorry, the pink colony on McConkey. Okay, this is another medium called mannitol salt agar, MSA. Okay, basically mannitol salt agar show the yellowish color, but Staph aureus show up yellow on the yellowish color. Everywhere Staph aureus grow, they process acid. They metabolize mannitol. Mannitol is sugar, okay? Metabolize acid and tolerate salt. That's MSA, selective media for Staph aureus. Sometimes we need to use McConkey, but generally when doctor observe Staph aureus, we observe from the other point, the other part of body. We not go to culture in McConkey except the food poisoning. Okay, special thing of Staph aureus, they tolerate a lot. When you isolate Staph aureus from the pus ก็คือหนองนะ the dry pus after 2-3 months you still can grow bacteria from the dry pus, so that is very tolerate. Okay, they can tolerate sodium chloride up to 10%. Okay, tolerate crystal violet and billion green. Both of these are bactericidal that can kill bacteria, but they are okay if the concentration is less than 1 in 500,000. That's 2 ppm. They tolerate 1% phenol for 15 minutes, and it's quite a lot. So the virulence factor of Staph aureus. Staph aureus has lots of weapons to attack us. Some weapons are secreted from the cells. Some weapons remain in the cell surface. Okay, let's start from the cell surface. We call membrane bound factor. On the surface, they have plasma membrane, peptidoglycan, and some strain, like I said, process capsule. Okay, capsule protect phagocytosis. Sorry?
แม่งเดี๋ยวกูเรียนอีกสักพักเลยว่ะน่าจะบ่าย 3 อ่ะ
Capsule inhibit phagocytosis. Okay. Peptidoglycan can activate immune response. They also have penicillin binding protein, PBP, all right, and protein A. I will say later what is that, and also some protein called adhesin help staph attach to the cell of host. Okay, secreted factor might be enzyme and toxin. Okay, invasive is classified as a type of toxin. Okay, invasive. Well, I would say Staph aureus comprise membrane bound and secreted. Let's start from the first one, capsule, membrane bound. On capsule, if they process capsule or slime layer, they probably prone to build biofilm and inhibit phagocytosis of macrophage, okay, and also neutrophil. Peptidoglycan itself can activate complement, can activate interleukin-1 and chemotaxis for PMN. PMN is neutrophil. Activate complement, activate IL-1. That means they can make your body inflammation. Lipoteichoic acid can bind to fibronectin. Okay, and protein A. Protein A actually is a blue part that can bind to FC region of IgG. Once the FC region, the circle is means Staph aureus. FC part of antibody bind to the cell of bacteria. That is opposite side of antibody should bind, right? So this is inhibit opsonization. The opsonization, that means antibody bind to bacteria, and white blood cells are eat bacteria easier because white blood cells have FC region receptor. But this is opposite. So this makes Staph aureus survive in your body. There are lots of enzymes from catalase. Catalase digest the hydrogen peroxides, coagulates that when when you get infected by a knife, cut your skin or whatever, get your wound. Staph aureus are normal flora on skin. They potential to go deep inside your skin. Okay. Staph aureus will produce coagulase enzyme and send out and make fibrinogen chain to fibrin and clump. Once it clumpy, that means they have protection around Staph aureus. At this time, white blood cell cannot reach to bacteria, and bacteria have time to multiply themselves, to increase in number. Once they increase in number enough, they use another enzyme called hyaluronidase. Because your skin, different cells of skin attached by hyaluronic acid. So hyaluronidase will digest hyaluronic acid and let bacteria pass through the tissue, go deeper in your skin. Okay. Lipase enzyme of Staph aureus also let them digest fatty acid on your skin. This allows Staph aureus spread from one location to another location. Fibrinolysin also lyses fibrinogen. When they get infected, they use coagulase to make fibroblast, the fibrin, protect the bacteria, and use staphylokinase digest that, go out, hyaluronidase digest tissue and go deeper in the skin. That's mechanism that Staph aureus try to attack you. And then they also release cytolytic toxin. This is a group of toxin. They comprise alpha hemolysin, beta hemolysin, gamma hemolysin, delta hemolysin. Okay, mostly destroy red blood cell. Okay, and leukocidin. Leukocidin, sometimes we call pentovalentine toxin, PVL. This toxin direct damage to PMN and macrophage. Is macrophage cell lysis, neutrophil cell lysis, so you have no innate immune response. Okay, that's the toxic of Staph aureus. Staph aureus also produce another type toxin called enterotoxin. Enterotoxin present in some Staph aureus, but if they release enterotoxin, they probably prone to cause the food poisoning when you eat food with contamination of toxin. Particularly in Thailand, you need to eat the food that just really cook by the proper cook, not just cook for a long time and leave it for a long time. Staph aureus probably contaminate in the food and produce toxin, and toxin contaminate in the food. The toxin can differentiate by the typing of antigen to be 9 antigenic type A, B, C, D, E, F, G, H, but C1, C2, C3, and I, whatever. But most virulent is toxin type A. Okay, this toxin resists the boiling for 10 minutes. So you need to use the heat and longer time for heat the food. They love to contaminate meat. Meat mean beef, pork, poultry. Well, poultry is not a major. Beef, pork, fish. Sorry, this is wrong. Milk and milk products. Okay, when you drink milk, you probably need to drink the UHT or pasteurization or something like that. And there is a super antigen. Okay, super antigen is mean when bacteria get infected, antigen presenting cell will engulf bacteria and digest inside that and present to MHC class 1 or 2. Well, extracellular is past MHC class 2. And MHC class 2 will go to trick with the T cell receptor, still remember? But superantigen is by outside of the group. The group is tricked together with T cell receptor and APC. But superantigen by outside, but can have ability to induce T helper, release interleukin 1, TNF alpha, interleukin 2, interleukin 6, a lot of chemokine, cytokine to induce inflammation. Okay. Enterotoxin is superantigen. Another toxin, also superantigen, called toxic shock syndrome toxin or TSST. This is very important for Staph aureus. You probably need to remember TSST. Okay.
Is that the toxin in what was that, the female products that used to have warnings that about toxin shock syndrome?
Pardon me? I didn't hear you properly.
Is this the toxin that the people were talking about in feminine products?
No, that is this one. Oh, yeah.
Oh.
If you talk about the use of the tampon, right? Use of something like that.
Yeah.
Yes, this is TSST. But I'm afraid you confused. But now you're not confused because we're just talking about this in the first time. In Streptococcus also produce this toxin. So then you probably differentiate when we talk about staph or strep. But now don't worry, we talk about Staphylococcus. Okay?
Yeah, okay.
Another toxin called exfoliative toxin or ET. Exfoliative toxin causes the disease called 4S. Staphylococcal scalded skin syndrome. Okay, let me, you just know the name first. Let me show you by process. The circle is your body. Okay, Staphylococcus locate. This is mouth. Your mouth, your nose, somewhere here. When you talk with someone, you probably generate small drop all the time when we use the mask cover to protect the virus for COVID-19, that also protect yourself from saliva droplet of the other person that might carry Staph aureus from that one to you. Okay, you probably inhale the small droplet, and then Staph aureus go to localize in your nasal. Okay, probably grow deeper. Okay, if you get infected by skin or they go by wound, if you have lesion on your skin, Staph aureus that live normally on your skin can penetrate the wound and get abscess. Okay, and you might get infection by touching your friends or someone else abscess or lesion. That is the one. Okay. Or they go deeper in the blood and produce TSST type 1 here, toxic shock syndrome toxin 1. Okay, and then you probably get a severe symptom from TSST, and staph also localized in the mucous membrane in vagina. That is the normal habitat of Staphylococcus just aureus. Okay, they transmit person to person. Okay, no vector. You free from mosquito bite. You just touch people who get infected. Indirect contact by aerosolites. someone sneezing, someone talking and saliva spill out, okay? You probably get endogenous strain by go deep in your body by operation with the unclean equipment, or once you try to recover from the operation, you might get infected after that. Accident, broken leg, broken arm, probably get infected by Staph aureus. And some people get infected without any symptom and signs. We call a asymptomatic carrier, but that guy can spread S. aureus to the other person, okay? So factor that make S. aureus tolerate immune is protein A. Protein A inhibit opsonization, all right? Coagulates make the blood clumpy and protect phagocytosis of PMN, okay? Hemolysin destroy red blood cell, destroy neutrophil, destroy macrophage, and even platelet. Leukocidin, this is a toxin destroy macrophage and neutrophil. They have penicillinase to inactivate antibiotic. They have several types of penicillinase. Okay, they have transpeptidase. Okay, that inhibit penicillin, also present in MRSA, methicillin-resistant Staph aureus. Okay, that's factor to disable immune system. But some enzyme can damage your tissue by hyaluronidase. Staph aureus need to go deeper in your skin by hyaluronidase. Okay, staphylokinase destroy the fibrin clot. Lipase destroy the lipid in your skin, let them spread out, make the wound bigger and bigger. And protease destroy protein. You probably heard some news on newspaper that we found bacteria that eat human meat, but actually that refer to Staph aureus because they produce protease and lipase, digest your meat and tissue. They produce toxin. Toxin is very harmful. They go together with blood and affect the other far away from the infected site. Exfoliative toxin cause 4S, okay? Staphylococcal scalded skin syndrome, SSSS, okay? Enterotoxin cause food poisoning. When you get contaminated food, you probably get a lot of nausea and vomiting and diarrhea, okay? TSST, toxic shock syndrome toxin type 1, cause staphylococcal toxic shock syndrome, okay? The disease from toxic shock syndrome is a broad symptom. You probably get pneumonia, infected in your lung because they contaminate your nostril. If you go deep in your lung, they're prone to get pneumonia. Or if you go to the blood circulation, you're prone to be meningitis. So disease from Staph aureus is quite broad. Osteomyelitis, you probably get inflammation of your bone, okay? Acute bacterial endocarditis, we talk about that in the next 2 slides. Make your heart muscle inflammation. Septic arthritis, make your joint pain. Skin infection, this is very simple and very normal, okay? Bacteria, sepsis. Sepsis mean you get infected by bacteria inside the blood circulation. In Thai, you probably heard the term ติดเชื้อในกระแสโลหิต That's sepsis. And if you get infected in urinary tract infection, particularly female, you might get bacteria might move up to the urinary bladder. Okay, the cutaneous infection by skin, you might get folliculitis. Folliculitis is mean as an abscess that happen by the, how can I say, the hair follicle. So everywhere you have hair, the root of hair probably get accumulation of Staph aureus and develop a pus. Pus ก็คือหนองนะ pus, the white part is pus. The white part of pus actually mean the white blood cell that came to destroy bacteria. Okay, stees mean infected on the hair follicle also, but happen in your eye, upper and lower lid of your eye. That is ตากุ้งยิง Okay, impetigo develop of honey skin, honey color on skin, kind of scale on skin, particularly happen on face around the mouth. That is impetigo. But not just only young children, but adult can be infected, but mostly in young children. And abscess. Abscess is ฝี You can be very small abscess until the very huge abscess. And when you get abscess, I'm sure you guys, no one can be free from abscess at least one time in your life. And you try to squeeze, use your fingers, squeeze the pus out. This is not a good idea, okay? You probably go to see doctor and let doctor cut the skin and release the pus outside. Because when they accumulate like abscess, that means Staph aureus accumulate underneath your skin, okay? And they cause fibroblasts cover the Staph aureus. If you apply pressure to squeeze that, fibroblasts probably break out inside your body, not another side of the skin. And that might spread to the internal organ, and you might get severe and prone to die. This is serious. Acute bacterial endocarditis is mean bacteria that Staph aureus, well, when you get infected in the blood circulation, Staph aureus probably go through and out and in and out all the time of your heart, and they accumulate. If your valve of heart has abnormal, just some people, allow bacteria localize and multiply itself on the heart valve, that might make the heart valve not close it properly. And when the doctor observed the sounds of heartbeat, they might see they might hear the noise called murmur. Okay, the murmur noise. So when doctors say they curious, like this patient probably get infected by gram-positive cocci, well, we point out Staph aureus. You need to take blood sample, 2 arms on the same time, from left arm and right arm, 2 samples. Because if you take just only 1 arm and you culture Staph aureus positive, you probably get contamination from skin. But you need to take 2 arms and get the same result. That's still like, draw conclusion is Staph aureus. The exotoxin of Staph aureus can cause food poisoning, and they normally contaminate with the protein-rich food and lipid-rich food in the United States of America. They contaminate it in mayonnaise quite often. So lipid-rich, be careful on mayonnaise lover, okay? Toxic shock syndrome can cause fever, can cause rash, can cause nausea, vomiting, or septic shock. Septic shock is mean like you get infected in the blood circulation and develop SSSS. The toxic shock come together with the long time use of tampon. Well, the tampon fiber can let bacteria bind to the fiber very easy. So don't use it for a long time. Okay. The disease of the toxic shock syndrome is make your skin peeling. Okay, like this in adult skin peeling, particularly by hand palm. But they cause disease in we call the systemic system, like they cause from headache, fever, watery diarrhea, vomiting, everything, quite a lot. Just no need to remember all, but no, you need to know that toxic shock syndrome is the key, one of the key syndrome of Staph aureus. Or in children, in young children, particularly newborn or children less than 10, we found SSSS because they infected and destroy the dermis, the epidermis, make the skin peeling out all over body. You see the red part of the skin. Actually, this is not rash, but it is the deeper skin because of the skin are peeled already. Okay. Treatment of Staph aureus. Treatment is just penicillin is drug of choice. If penicillin is not work, doctor probably think about methicillin or cloxacillin. They are methicillin group. But if you test the lab test, this is MRSA because methicillin resistant. Probably use vancomycin. For MSSA, methicillin sensitive, we use methicillin, that's okay. Okay. Wound drainage. If you get abscess, you need to drain the abscess. Okay. Device removal. I mean, so someone who put the device for birth control device. If you get device and you feel like irritation and get infection by Staph aureus, you probably need to remove that device. Or doctor need to remove the dead tissue because, well, bacteria destroy tissue. Okay. Prevention. Now we have no proper vaccine for Staph aureus. So just clean your hand. Keep the wound covered, okay, to protect the infection of the Staph aureus. In this case, we talk about Staph aureus. Reduce tampon risk, okay? That's avoid sharing personal care. Particularly female student. I don't know, you guys feel like keep, well, some of you probably take lipsticks or hair, a comb hair, towel, whatever, t-shirt when you go to stay your friend's apartment, working in group, and lots of students may go to the friend's room and don't use your friend's stuff because you probably spread Staph aureus to each other. And cooking and storing food properly, don't let contamination. So Staph aureus is very broad. You can get infected from the very small or easy disease until the very, very hard and life-threatening, like sepsis. Okay. Right. Now we move to Staphylococcus epidermidis. Staphylococcus epidermidis differ from aureus. Aureus mean golden. Epidermidis is mean like epidermis. They are normal flora on skin actually. Colony color is white, unlike Staph aureus. This one is coagulase negative, okay? But the other thing are similar to aureus. On McConkey agar, they show pink colony. On mannitol salt agar, they also show pink, not just like yellow as Staph aureus. The virulent factor, they produce slime layer instead of capsule. They prone to produce biofilm. Okay, biofilm protect the phagocytosis of PMN and macrophage. Okay, they mostly opportunistic infection for immunocompromised patient. Okay, then they are not infect you guys at home, but if you stay or admitted to hospital for a long time, you probably get infected by hospital acquired infection. Okay, that's easy for, well, I not emphasize much, but just let you know how different between epidermidis and Staph aureus. The key is Staph aureus. Okay, methicillin is also work well. Okay, Staphylococcus saprophyticus. Saprophyte mean the junk, the waste. Okay, the waste or rubbish. So this bacteria grow in the waste thing, waste substance. Okay. By colony is white on blood agar and no hemolysis. Okay. Coagulase negative normally cause urinary tract infection. Okay. So Saprophyticus and epidermidis differentiate by novobiotin disc. So novobiotin is a drug that can kill epidermidis. So epidermidis is susceptible to this drug, but Saprophyticus is resistant, so drug cannot do anything with this bacteria. Okay, just let you know, they occasionally get infected. Okay, mostly GI tract, get food poisoning. All right, summary. In summary, for gram-positive, you need to test gram-positive cocci arranged in cluster. Okay, catalase positive, you can say this is Staphylococcus only. But if coagulase positive, it is Staph aureus. Okay. If coagulase negative but novobiotin positive or sensitive, that's epidermidis. If novobiotin is resistant or negative, means Saprophyticus. That's the key. This is the conceptual key for diagnose Staphylococci. Next week you have a lot more. Okay, trying to find a very huge paper and then you just pick the note and draw the flow, and finally you get a very big flow and help you remember. All right, question? No question.
I will have just one question, but it's just out of curiosity. I understand all the biochemical tests to identify the species of bacteria. Can we do like the virus PCR? I mean, I guess we know the sequence and we can also analyze the genome to know which species it is, or we never do that?
Well, PCR detection also performed in Staph aureus, but PCR detection cannot tell doctor for the drug resistance. And some PCR can cross to the other staphylococci. So doctor want to know this is Staph aureus or other staph. The biochem still work. Just only the other bacteria, molecular come to overcome the conventional, but gold standard for bacteria to detect like this is the causative agent, you need culture. If you cannot culture, it's hard to claim this is a causative, right? Once you get sample, well, let's say if patient get sepsis, specimen will be blood. If patient will be abscess can be streptococcus, staphylococcus, whatever you learn later on. But if doctor curious, it should be staph, you get pus as the specimen. So you get so variety of specimen. If you get UTI, urinary tract infection, urine as specimen. If you get food poisoning, feces, stool sample as specimen. So dealing with Staph aureus is so variety. And this makes student headache a lot. So that's why I pick up first. So you have time to read and keep asking. But there are some difficult bacteria later on, but Staph is, if you practice in the lab and you want to be a scientist in the future, you're working with the medical section. Staph aureus is one of very few bacteria that is very impact. So I think learn something impact first, and less impact, it depends on your passion. Okay, but if you're working like, oh, I don't know, I never know about Staph aureus, you kind of fail. But you don't know some rare bacteria, that's okay. But Staph aureus, I'm sure once in your life you get infected by small abscess, by pimple, by whatever, you probably get a small piece of wood or small piece of the pinch your finger or something, you get pus, even so small, that's Staph aureus. Then you know the mechanisms, like how Staph aureus overcome your immune system. They protect by fibrin shield. Immune cannot go inside. Once they're ready, they use another set of enzymes, destroy the shield, come out, and immune shock like, wow, you have a lot. In the beginning, you come just like so few bacteria, then they protect themselves. And they try to escape by spend hyaluronidase, go deeper, try to reach the blood circulation. Once they reach blood circulation, that means they go everywhere. So the disease kind of more serious. This is Staph aureus. But if you understand Staph aureus, Streptococcus kind of easier. They are something similar. So if you have time this weekend, review of Staph aureus. Okay, and next week we'll learn the other gram positive. We'll finish gram positive next week. Okay. Some more questions? Oh, next time, probably have lunch before class, because we have so many pictures that make you cannot, don't want to have lunch after class. All right, so then see you next time on Thursday. See you.
Thank you so much. See you next week.
Thank you so much ค่ะ
สวัสดีครับ ขอบคุณครับ